Chapter 8 is covering microbial genetics. Genetics is the study of inheritance or heredity of living things. How do you get the characteristics you have? For bacteria, how does a bacteria know that it's supposed to be gram positive? How does it know it's supposed to be gram negative? How does it know how to put together the cell wall so that it is gram positive or gram negative? It's all inside of the genetics. Genetics simplified are our instruction manual, or I should say are the bacteria's instruction manual. The field of genetics is going to examine the transmission of biological properties called traits from parent to offspring. The traits in bacteria could be antibiotic resistance. It could be, again, the gram negative or positive nature of the cell wall. Or is it a mycobacterium where it has an acid-fast wall? The expression and variation of those traits. In human terms, this might be instead of having blonde hair or brown hair, you have red hair. What's the variation? The structure and function of the genetic material. How does it work? What does it look like? How the genetic material changes. We talk about mutation quite often, and mutation is literally the changing of the genetic material. How does it get altered from one generation to the next? In humans, it's a lot more complicated because we're not making clones, we're making unique individuals. So it's a bit easier to see how you can shuffle things around and get something unique. With bacteria, it's a bit different, but we'll get to that. The genome is all of the genetic material of an organism, the entire library that is contained inside of the DNA. It usually exists in the form of chromosomes made of DNA. Viruses can have a genome. That's how it knows how to make the capsid, how to take over the machinery of the cell. But the big, glaring difference with viruses is that genome, that instruction manual can be RNA or it can be DNA. You don't see that anywhere else. Everywhere else, it's always going to be DNA. Viruses are unique in that way when it comes to the genetics. Bacteria can also have extra chromosomal elements, genetic material that is not part of the chromosome. These are called plasmids. These are those extra rings of DNA that I had previously mentioned. Plasmids contain genetic information for special characteristics such as antibiotic resistance. Let's talk terminology. A gene. It is a specific segment of DNA that contains a code, an instruction manual, to make a protein, or it codes for a trait. So I need a channel, a doorway, for sodium to get into my cell. There is a gene for making sodium channels somewhere inside of my genetic code. Again, it's an instruction manual that tells me how to put things together. Codes for a trait. I have long fingers. Somewhere in my genetic code, there is a trait for long fingers that when I was developing in the womb, actually said, this is how your fingers will look. And my cells said, okay, those are the types of fingers I will build, because the instruction manual said that. We have two primary types of genes that we discuss: structural genes that code for proteins and for traits, and regulatory genes. Regulatory genes are exactly what they sound like. They regulate. In this case, they are regulating gene expression. They control when these things happen, when you see them. So literally, I am controlling the gene's expression. Does it get expressed or does it not get expressed? I'm the one to decide that. Genotype versus phenotype. Genotype is the sum of all genes in an organism, the distinctive genetic makeup of an individual. Phenotype is the expression of the genotype, usually the characteristics that we can see. So genotype is the actual code. What does it say? It doesn't tell you what it does. It's just, here are the words. Okay? So genotype is adenine, thymine, uracil, adenine, thymine, cytosine, guanine, cytosine, thymine, guanine, cytosine. That sequence is the genotype. What does it say? Phenotype is, when I'm looking at you, what do I see? So the sequence may be adenine, thymine, guanine, cytosine, whatever, but that instruction manual is for long fingers. So the phenotype would be long fingers. The genotype would be the actual sequence itself. A nucleotide is the basic unit of DNA. So if I were to talk about Legos and building a castle out of Legos, the basic unit would be the Lego. Just one Lego all by itself. Now I can put them together into this complex structure. I can build the Death Star if I want to, but the basic unit would be the individual Lego. DNA and RNA are made out of this basic unit called a nucleotide. It's made up of a phosphate, a deoxyribose sugar, and a nitrogenous base. Now, deoxyribose is specific to DNA. If we were talking about RNA, this sugar would now be a ribose instead of a deoxyribose. And literally the difference, deoxy, take out an oxygen. With ribose, you have the oxygen. With deoxyribose, you don't. Nucleotides link to each other to form the backbone of a DNA structure. Okay, or a DNA strand. So if you look here, which I will show you better in just a second, there's a ladder that looks like it's twisted. The sides of the ladder, well, I should do it this way. The sides of the ladder between the, I should say on the sides of the rungs are the backbone. This is what holds everything that is interconnected in the middle. So that backbone, okay, links the nucleotides to each other. So rung to rung, they're connected by that backbone, okay? You have things called complementary base pairing, okay? So complementary base pairing basically means that you have a partner that you always go to. In the case of DNA, A always bonds with T and G always bonds with C. Okay? So for DNA, that bonding, which you can actually see here, there's G and C, there's A and T, and you'll notice G and C are always together, A and T are always together, is essential in the fact that you always have to have the right partner. That complement, in other words, you're with the right partner, ATGC. Now, if you're from Alice, Texas, you can think Alice, Texas, go Coyotes. And think about how to spell coyotes to kind of remember that A and T go together and G and C always go together. That complement ensures that your DNA has the right spacing between it. When you have the correct partners together, you have parallel sides. Literally, they are straight. If you have the wrong partner, one of two things will happen. Either it will bulge out like this, which means that your ladder goes, meh, or it'll basically push in like this. So your ladder goes from being straight to kind of curving in and then going back to straight. You don't want the wrong partner. When you do, it doesn't sit correctly. And for your cell, that is a big, huge red flag that something wrong is happening in that area. Let's take these images and blow them up. Here, you should see that our DNA has this twisted ladder shape that we call a double helix. Your base pairs, okay, I should say your nucleotide. You've got a 5-sided sugar, okay, so that's the sugar. You've got a phosphate group, and then on this side, you have your nucleic acid. So for DNA, well, actually, let me put this here, DNA, your sugar is deoxyribose. Your nucleotides can be adenine, thymine, cytosine, or guanine. All right? If, however, we were talking about RNA, the sugar would be ribose. And while most of the nucleic acids would be the same, this one is different. So this one would be uracil. Uracil is unique to RNA. Thymine is unique to DNA. Wow, I really misspelled ribose. Ribose. Jeez, can't take me anywhere. The reason that I make the distinction primarily is so that if you get a sequence on a piece of paper, the minute that you see uracil, you can automatically identify that you're looking at RNA. The minute that you see a thymine, it's, again, automatic, you're looking at DNA. The unique factor of those two nucleic acids means that you can, just by looking at sequence, determine what you're looking at. Sugars are a bit more difficult because when it comes to looking at sequences, usually they're not going to say anything about, oh, there's the sugar. They don't really do that, but they will give you the nucleic acid sequence. And if you can see a uracil, if you can see a thymine, you know which one you're examining. Moving on from there, here's the phosphate backbone. You can see that I've got phosphates all the way down the sides of my ladder. You can see the sugar connected to that phosphate. There's the deoxyribose. And the rungs of my ladder are being created by the bonds between my nucleic acids. Something to note, adenine and thymine, anytime that you see them, have 2 bonds between them. Cytosine and guanine will always have 3. This is another reason why when you have the wrong pairing, it looks like a bump or a dip in the ladder instead of them being completely parallel to each other. if I'm looking for somebody to partner with 2 bonds, and I have somebody to partner with 3, it doesn't fit the right way. It would be equivalent to having a 3-prong plug and trying to fit it into a 2-prong outlet. It doesn't fit correctly.